Hola, it is an honor and a pleasure to present to you our work entitled Facing Long-Branch Anxiety and Outgroup Prejudice in the Phylogenomic Analysis of Flaviviridae. We divided this presentation into 5 sections. We will introduce to you a family of viruses called Flaviviridae, especially its genus Flavivirus. We will explain why we are interested in the annotation and phylogenetic analysis of these viruses, and we will introduce the specific questions we are trying to answer with this work. To answer those questions, we will show you a new approach for the annotation of the genomes of flaviviruses and a series of methods to analyze how that may impact phylogenetic analysis. We will end this presentation with the discussion of our results, a few final remarks, and information on supplementary digital materials. Introduction. The family Flaviviridae is composed of Flavivirus, Hepacivirus, Pestrovirus, and Pegivirus. These viruses are enveloped, spherical, and have about 15 nanometers in diameter. Their genomes are single RNA molecules that encode for a single polyprotein with about 12 protein-coding genes inside. The gene contents varies among genera. Viruses in this family, including dengue, Zika, hepatitis C, West Nile, and yellow fever, pose an ongoing and severe threat to public health. To respond appropriately, it is vital that we understand how these viruses evolve, emerge, and spread. Central to this understanding is an updated phylogeny for the entire family. Despite the relative simplicity of their genomes, only 60% of the sequences at NCBI's databases are annotated beyond the polyprotein level. Our bibliographical revision indicates that annotation is not a significant concern for most authors. The majority of papers align the entire polyproteins. Then, due to the variation of gene content among genera and also a non-Pellin declared fear of long branch distortion caused by distant sequences, most authors perform phylogenetic analysis and present the trees with midpoint rooting. This approach, however, is problematic in many ways. For example, the alignment of complete polyproteins may result in nucleotides from different genes aligned to each other, which could be an indication of false homology statements. Furthermore, midpoint rooting may create artificial clades, and the lack of outgroups may hamper the correct assumption of the order of character transformation. Therefore, we made ourselves 4 questions. First, can we efficiently annotate complete genomes of Flaviviridae using reasonable amount of resources? Second, does the analysis of partition genes result in different hypotheses than the complete analysis of polyproteins? Third, will outgroup sampling significantly impact hypotheses of in-group relationships when we have data from complete genomes? And finally, does long branch distortions frequently impede the inclusion of outgroup data? A new approach for viral genome annotation. We analyze the genome through this pipeline, starting with gene predictions using GeneWise and transdecoder. With the help of BLAST and Hammer, we combine homology-based and ab initio annotation strategies. We pooled the alignments and calculated distant matrices for outlier testing using the Tukey method. Finally, we removed all outliers. This pipeline is currently available in GitLab and relies on open-source software. However, our team is preparing a web application to facilitate usage. Now for the other methods. We selected 78 complete reference genomes to evaluate the quality of our annotation strategy and another 41 non-annotated genomes. The final dataset had 103 flaviviruses and 16 outgroup sequences. Given the different gene content of each group of viruses, we decided to partition the data in 14 sections as we show here. First, we build 3 matrices, one with all the terminals and all the partitions, another with partition sequences of flaviviruses, and the last with non-partition data of flaviviruses. We use different strategies to account for the effect of sequence alignment. Likewise, we performed research under parsimony, maximum likelihood, and posterior probability to assure that the observed effects were not due to research strategies alone. The protocol above resulted in 31 tree search experiments, and we calculated the match split distance among all unrooted trees generated this way. To test the sensitivity of trees to partitioning and outgroup sampling, we pruned them so they would have the same set of terminals and calculated the distance among the pruned trees. We also removed the longest branches we found in a series of systematic interactions and reanalyzed the data under parsimony to see if we could detect any long branch distortion. Results and discussion. First, the annotation pipeline worked with maximum efficiency for most genes, especially those that are prevalent among the different genera. We observed no false positives, and we annotated all 119 genomes in less than 2 hours. A clustered analysis of different cladograms, organized by their distance, shows that outgroup sampling and partitioning together generated trees that are similar to each other and different from all others. The dendrogram indicates that the inclusion of outgroups allied with the annotation of the genomes significantly impact tree search experiments. A visual inspection of all the cladograms suggested that the trees that did not come from the complete and partitioned datasets would frequently disagree with groups of viruses that are almost a consensus in the specialized literature. We evaluated all the trees manually and did not recognize any distortion caused due to long branches. It is worth noting that trees from which we pruned a long branch are different from the original trees. However, the distance among the trees from which we pruned a long branch is similar to the variation that is found among the original trees. We expected this result since any terminal can potentially impact tree topology, but if any of the removed terminals were responsible for a long branch distortion, we would expect a significantly difference between the resulting topology and the other pruned trees. Since that was not the case, we are assuming that no long branch distortion occurred. Here we show the updated phylogenetic hypothesis for Flaviviridae. As the working hypothesis, we selected the tree topology from a complete and partitioned matrix aligned with a translation-based method and searched under maximum likelihood. We recovered the monophily of all genera. This figure focuses on the passivirus, pegivirus, pestivirus, as well as their relation to flavivirus. Among the most important things to note here is the absence of the soybean cyst nematode virus 5. This sequence was flagged as an outlier by our pipeline. The soybean cyst nematode virus is much larger than all other Flaviviridae, and data from its original publication already shed some doubt over its position within flaviviruses. And here we confirm that the soybean cyst nematode virus 5 is not a member of this family. Also worth noting is the position of Tamanabat virus within other flaviviruses as a sister group of ESFV-specific lineages. There was some debate over its taxonomy since it was very long in comparison to other branches of Flaviviridae. But our analysis confirms that it belongs to Flavivirus. Flavivirus would become paraphyletic if we classify Tamanabat virus as a different genus of viruses. This figure presents the relationships among flaviviruses, mostly related to tropical diseases transmitted by Aedes and Culex, including dengue, West Nile, yellow fever, and Zika virus. Overall, the tree corroborates the major groups of flaviviruses according to recent revisions of the genus. Despite agreeing to the main clades in recent revisions, our analysis brings additional insights to the phylogenetic relationships within each of them. We draw attention particularly to the internal relationships of the clade Aedes species-associated MBFV2 in this figure. Our analysis places dengue and Zika in a much closer relationship than previously observed from non-partition analysis of flaviviruses alone. Now for the main conclusions. The annotation is efficient and fast. We observed no false positives, and the pipeline runs in a few minutes on a personal computer. The outlier removal flagged the soybean cyst nematode virus 5, adding to the hypothesis that it does not belong to Flaviviridae. Our team is currently working on a web application to facilitate the usage of the pipeline. The effect of partitioning on trees is significant, and certain parts of the tree space were more accessible for partitioned data that contained outgroups. Also, partitioned analysis agreed more with the specialized literature, providing resolution to uncertain branches, such as the relationship between Zika and dengue viruses. Outgroup sampling also had a profound effect on tree topology, although it requires complete gene annotation. Otherwise, spurious homology statements, especially when different genera are analyzed together, could diminish the quality of the proposed tree hypothesis. We did not observe distortions due to the different branch lengths. We argue that long-branch distortions, such as long-branch attraction and parsimony, is easy to test by systematically removing long branches and analyzing its effects, which can be done using protocols such as the ones we applied in this work. As a final remark, we argue that preoccupation with long-branch distortions is valid. It justifies result analysis, but it does not justify the avoidance of outgroups. We are thankful to the University of North Carolina at Charlotte and the University of California, San Diego. We also appreciate the computational resources made available to us at the University of São Paulo in Brazil. The QR code in this URL will take you to a website with the presentation and supplementary material. Thank you.